This lecture explains the transcriptional repression of genes through methylation of cytosine within CpG sequences, as well as the demethylation reaction. In animal cell genomes, majority of base modifications observed are methylation of the fifth carbon of cytosine within CpG sequences. In 5-methylcytosine, the hydrogen at the fifth carbon position of the cytosine base is replaced with a methyl group. This reaction is carried out by DNA methyltransferase. In double-stranded DNA, CpG sequences are palindromic. When a CpG sequence on one strand is methylated, the corresponding CpG on the complementary strand is also typically methylated. Methylated CpG sequences are inherited in DNA strands after genome replication. Additionally, methylated CpG sequences can induce histone modifications, leading to chromatin condensation, which can stably suppress the transcription of specific genes over long periods. The enzyme that catalyzes the methylation of cytosine is DNA methyltransferase, abbreviated as DNMT. The methyl group donor used for the transfer is S-adenosyl methionine. S-adenosyl methionine is a complex of the amino acid, methionine and ATP, as shown in this figure. The cytosine content in the human genome is approximately 20%. The methylation of cytosine in CPA, CPT, and CPC sequences is almost zero. On the other hand, the methylation of cytosine in CpG sequences is about 70%. However, the methylation rate varies greatly, depending on where the CpG sequence is located within the genome. In the region near the gene promoter, spanning 500 to 3000 base pairs, there are more CpG sequences than in other areas, so this region is called a CpG island. Limited to these CpG islands, the frequency of cytosine methylation is low, only about 10%. In CpG islands, the frequency of cytosine methylation is generally low, but variations are still observed. Genes involved in maintaining basic cellular functions are called housekeeping genes. Examples include actin genes, ubiquitin genes, and ribosomal RNA genes. In these housekeeping genes, which are always highly transcribed, the methylation rate of their CpG islands is almost zero. On the other hand, non-housekeeping genes undergo methylation during development and differentiation. Additionally, in cancer cells, the frequency of methylation in CpG islands of tumor suppressor genes is often high, resulting in suppressed transcription of the genes. It has been observed that the occurrence rate of CpG sequences in the genome is significantly lower than the expected rate, calculated from the proportion of cytosine in the genome. The human genome consists of approximately 3 billion base pairs. Since the occurrence rates of cytosine and guanine are both 20%, the estimated number of CpG sequences in the genome is 120 million. However, the actual number of CpG sequences is only 32.28 million, which is merely 1/4 of the expected number, based on probability calculations. This suggests that there has been an evolutionary pressure in the human genome to avoid CpG sequences. One possible explanation for this evolutionary pressure is that 5-methylcytosine is prone to demethylation within cells, which can lead to its conversion to thymine. If this thymine is not removed before genome replication, it can cause mutations. Let's look at base modifications in eukaryotes, other than humans. In plants, in addition to CpG sequences, cytosines within ChG and CHH sequences can be modified to 5-methylcytosine, similar to animals. On the other hand, in fungi such as yeast and mold, base modifications are so low that they are barely detectable. Also, In nematodes, there is no methylation modification of cytosine, but adenine undergoes methylation. I will now explain the effects of methylated cytosine on transcription. When cytosine in the promoter sequence is methylated, it reduces the binding affinity of transcription factors to the promoter, as shown here. This results in the suppression of transcription frequency. On the other hand, once transcription has started, the presence of methylated cytosine within the gene body does not hinder the progression of transcription. Furthermore, methylation of cytosine induces specific histone modifications. These histone modifications promote chromatin condensation, leading to heterochromatinization, which prevents transcription factors from binding to DNA. And it results in stable gene expression suppression and transcriptional repression. This robust suppression of gene expression, formed by the coupling of DNA modification and histone modification, is called epigenetic repression. The term epigenetic refers to the phenomenon where genes behave as if they have undergone mutations, even though no actual changes have occurred. Modified bases can also be found in prokaryotic genomes. These modifications include methylation of adenine and methylation of cytosine. Classification of DNA methyltransferases in bacteria. Restriction-modification RM system. Bacteria possess restriction enzymes within their cells to prevent the invasion of parasitic factors, such as phages. To protect their own genome from being cut by the produced restriction enzymes, bacteria also have DNA methylation enzymes that methylate cytosine, or adenine, within target sequences. For example, EcoRI methyltransferase methylates adenine in the GAATTC sequence, preventing the restriction enzyme, EcoRI, from cutting this sequence within the bacterial genome. The second type of methylation enzymes are not part of the restriction modification system and are called standalone or orphan methyltransferases. Well-known examples include DCM methyltransferase and DAM methyltransferase. These enzymes introduce methyl modifications to cytosine or adenine within specific sequences, but there are no corresponding restriction enzymes that cut these target sequences. I have already explained the role of methylation enzymes in the RM system. As for the role of orphan DNA methylation, it includes the regulation of gene expression levels. Additionally, it plays a role as a signal for distinguishing between the old strand and the newly synthesized strand in the repair of mismatched bases. I will explain the mechanism by which methylated cytosines within CpG are newly maintained, even after DNA replication. Before DNA replication begins, both strands of the CpG sequence are methylated. This state is called the fully methylated state. When DNA strand replication occurs, the methylation of CpG on the old strand remains intact, but there is no methylation on the CpG of the newly synthesized strand. This state is called hemimethylation. There is a DNA methyltransferase that methylates unmethylated CpG sites on the newly synthesized strand, which is named DNMT1. DNMT1 is an enzyme that introduces methylation to cytosines and change hemimethylation sites to full methylation sites. So it is called a maintenance methylase. In addition to such maintenance methylases, there are also methylases that introduce methylation to cytosines that have not been methylated before, and these are called de novo methyltransferases. I will explain how the maintenance methyltransferase DNMT1 fully methylates hemimethylated sites. There is a slight difference in the mechanism between early S phase and late S phase of DNA synthesis. First, let's discuss the non-replication coupled methylation that occurs in late S phase, which is not simultaneous with DNA replication. It would be simple and easy to understand if DNMT1 recognized hemimethylated sites and methylated DNA, but this is not the case. UHRF1 is a protein that binds to hemimethylated CpG sites. When UHRF1 binds to a hemimethylated CpG site, it ubiquitinates lysine residues near the N-terminus of nearby H3 histones. The ubiquitination recruits DNMT1, which then methylates the unmethylated CpG. The ubiquitination of histone H3 by UHRF1 plays a crucial role in facilitating the maintenance of DNA methylation patterns during cell division. Next, I will explain the methylation of newly synthesized DNA strands, coupled with DNA replication, during the early S phase. In this case, UHRF1 ubiquitinates the N-terminal lysine of the PAF15 protein, which is part of the DNA replication complex, rather than the H3 histone. This ubiquitination recruits DNMT1 to the PAF15 protein, AAA, which then methylates the unmodified CpG. This is the replication-coupled CpG methylation, observed in the early S phase. So far, I have explained the function and maintenance mechanism of methylated CpG. Now, I will also explain the mechanism of demethylation of methylated CpG. There are two mechanisms for demethylation of methylated CpG, passive and active demethylation. Passive demethylation occurs when the DNMT1 enzyme does not function, resulting in no methylation in the newly synthesized strand. When the DNMT1 enzyme does not work, the number of unmethylated CpG sites increases with each DNA replication. This occurs for the maternal genome in early development. On the other hand, active demethylation is achieved by active oxidation of 5-methylcytosines, which are then removed by base excision repair. In animal cells, this occurs during spermatogenesis or in the paternal genome during early development. This data shows the analyses of CpG methylation for the OCT4, NANOG, and X-ISD genes in white blood cells. On the other hand, sperm is a reproductive cell. Black circles indicate methylated CpG sites, while white circles represent unmethylated sites. In white blood cells, we can see that many of the CpG sites are methylated. On the other hand, in sperm, most CpG are unmethylated. This indicates that in cattle, extensive demethylation of methylated CpG occurs during the process of spermatogenesis. Active CpG demethylation occurs through base excision repair of mismatched base pairs. Here's an explanation of this process. 5-methylcytosine is prone to deamination reaction in cells, which convert it to thymine. As a result, the guanine with 5-methylcytosine base pair becomes a G with T mismatch. This mismatched base pair is then processed by thymine DNA glycosylase TDG, which excises the thymine base. The reaction creates an apyrimidinic site, AP site. After AP endonucleases and phosphodiesterases remove the phosphate group and sugar, DNA polymerase and DNA ligase insert deoxycytidine monophosphate, DCMP. This process effectively demethylates the methylated CpG to an unmethylated CpG. This mechanism demonstrates how the cell actively removes methyl groups from DNA through a series of enzymatic reactions, ultimately restoring the original cytosine base. This figure summarizes various modifications of cytosine base and the formation of mismatched base pairs. Cytosine is converted to 5-methylcytosine by the maintenance methyltransferase DNMT1. At this point, the base pairing between G with 5-methylcytosine is not considered a mismatch, so 5-methylcytosine is not excised. When the demethylation system is activated, 5-methylcytosine is converted to 5-hydroxymethylcytosine, 5-HMC, and further to 5-formylcytosine, 5-FC, by TET enzymes that are oxidative enzymes. The pairing of G with 5-formylcytosine, 5-FC, is recognized as a mismatched base pair, so 5-FC is excised by thymine DNA glycosylase, TDG. Subsequently, DCMP is inserted through base excision repair, resulting in a G with C base pair. If 5-FC is not excised, it is further oxidized by TET enzymes to 5-carboxylcytosine, 5-CAC. This is recognized as a mismatched pairing, and through base excision repair, and cytosine is inserted. In this way, 5-methyl-C is oxidized by TET enzymes to create mismatched base pairs, which are then removed by the base excision repair system, resulting in the demethylation of methylated cytosine to cytosine. This figure shows the changes in DNA methylation frequency during early human development. Unlike in cattle, in humans, demethylation has not yet occurred during germ cell formation. Extensive demethylation occurs during the first 14 days of development after fertilization. The maternal genome is protected by maternal proteins, so TET enzymes do not act on it. Also, since there is no DNMT1 enzyme inside the fertilized egg, a slow passive decrease in methylated cytosine frequency occurs through cell division. On the other hand, in the sperm-derived genome, methylated cytosine is oxidized by TET enzymes, forming mismatched base pairs, which are then removed, resulting in a rapid decrease in the frequency of methylated cytosine. Even during this early development, there are 100 to 200 genes that do not undergo demethylation, and these are called imprinted genes. The content of this video can be viewed in text and images on the home page site, biology website, Life Science Lectures for you. The site address is given in the description field of this video.
はい。ではこれで終了します。
If you have not found it yet, keep looking.